So the question may come up, why are we talking about all this internal, external segment, globus pallidus, caudate, etc., etc.? And the answer is, it's really important to understand what's going on in the normal system, right? The basal ganglia as it functions normally, because what we'll see is that when we talk about Parkinson's and Huntington's disease, that'll be a pathological state. So we have to understand how the system works normally, I think, before we can see how it works in the case of neurodegeneration. And so I just want to give you kind of a quick overview before we layer on another piece of complexity to this puzzle. All right, so the idea is that there's some sort of sensory input, right? And neurons that we consider ascending will work their way up toward the brain and eventually into the cortex, right? The cerebral cortex. The cerebral cortex will provide synaptic output, right, that enters the caudate and the putamen, right, the striatum. And when this happens, right, this is excitatory input, and so it causes the caudate and putamen to increase their output, but their output is inhibitory. Okay, and so if we focus on the globus pallidus, particularly the internal globus pallidus, the GPIS, what this inhibitory inhibition does is it shuts off the tonic inhibition, which the GPIS puts out normally, right? So this inhibition here is tonic. Okay, and if we have this level of inhibition here, we disinhibit this circuit, and it allows the VAVL complex of the thalamus to provide excitatory input to frontal cortex, right? And this leads to our motor response. So if we get activity in the medium spiny neurons, right, the MSNs that are found in the caudate and the putamen, if these neurons are generating action potentials and releasing GABA, that will lead to, even though GABA is inhibitory, that will lead to a motor response through disinhibition. The pathway that we didn't talk about much might just be worth mentioning here quickly, right? Again, we're going to have the same idea that we have cortical stimulus into the caudate and putamen, and in this case, right, the MSNs release GABA onto the substantia nigra pars reticula, the pars reticulata, okay? And so again, this is a disinhibitory effect where normally the SNPR, right, that's this thing here, is providing tonic inhibition to this superior colliculus. The superior colliculus is responsible for eye movements. And so if your eyes are focused in one direction, and they're not moving, it's because there's tonic inhibition of those motor control pathways. But when you need to move your head or your neck or your eyes to see something, right, the medium spiny neurons will be active. They'll release GABA. GABA will inhibit the neurons of the SNPR. That'll disinhibit the superior colliculus, and you'll have a motor response that allows your eyes to shift or your neck to move. Okay, but again, what we're going to focus on with respect to Parkinson's is this pathway here, this segment of it. We won't worry about the superior colliculus as much. So one other important point to make before we go on, right? We said that input to the striatum is driven by the cortex, right? And that this is glutamatergic. But that's not the only place that input to the caudate and the putamen can come from, right? We mentioned one other place. The substantia nigra pars compacta is a brainstem structure that can also send axonal projections and stimulate the caudate and the putamen. So if you go back to this figure here, right, what we see is the cortical pyramidal neurons which are synapsing on this medium spiny neuron somewhere in the striatum. But we also have this dopaminergic neuron, and this is going to become important later, right? And this dopaminergic neuron is typically a brainstem neuron, and it's in the substantia nigra pars compacta, okay? And it's going to release dopamine onto the caudate and the putamen. And we'll see what happens when we get to Parkinson's disease, but this is going to play an important role, so I don't want to forget about that. Taken together, right, everything together, this pathway that we just talked about makes up the direct pathway, right? So we've got two different inputs to the striatum here, right? There's the cortex and the pars compacta, right? So this is a brainstem. So the cell bodies are in the brainstem and their axons project to the caudate and the putamen. And these are cortical cell bodies and their axons project down into the caudate and the putamen. So the cortex, right? But they're both providing excitatory drive. That excitatory drive, again, inhibits the tonic inhibition here and allows these cells in the thalamus to provide excitatory input to the cortex, which results in movement. Okay, we've been over that now a couple times. But we have to talk about a second circuit, the indirect circuit, which plays a supporting role. So there's an additional circuit that makes up the indirect pathway, which helps to raise the level of tonic inhibition mediated by the globus pallidus internal segment here. Okay. So maybe we should note that. We can say that there's a second pathway besides the direct pathway. Which helps to raise the level of tonic inhibition. Oops. Of this indirect pathway. Oops, of the direct pathway, excuse me. In this case, medium spiny neurons are projecting to the globus pallidus, but the external segment, okay? So medium spiny neurons project to the GPES. Remember this diagram? Right, so you've got one pathway, the indirect pathway, would have projections from the striatum to the GPES, whereas the other pathway, the direct pathway, has projections from the medium, so this would be a medium spiny neuron in the striatum to the GPIS. And we're going to focus on this one, which is where we see the beginning of the indirect pathway. This is inhibitory input, and it's mediated by GABA, okay, just like to the internal segment. Axons projecting from the external segment target two different areas. They either target the internal segment of the globus pallidus, the GPIS, or they project to what's called the subthalamic nuclei. The projections from the subthalamic nucleus, right, those axons, synapse on the internal segment. Right? And this synapse is excitatory. In this place, glutamate is used as the neurotransmitter. Okay, and we know where the globus pallidus internal segment projects to, right? It goes on to the thalamus, which goes on to the frontal cortex, right? The motor pathway. Activation or stimulation of this pathway can occur in two different ways, right? The cortex, the cerebral cortex, right, that's received sensory information, can directly activate the subthalamic nucleus. This is a glutamatergic synapse. So excitatory output excites the subthalamic nucleus. And the subthalamic nucleus then releases more excitatory neurotransmitter onto the internal segment. Or we could stimulate the medium spiny neurons with the substantia nigra pars compacta, right? The substantia nigra pars compacta, just like we saw in the direct pathway, okay? So I want to point out the things that are in common, right? This is in common, and this is in common, okay? And both can lead to activation of the circuit. Let's focus on what happens when the indirect pathway is stimulated. So pre-stimulus, before the pathway is activated, the GPES is similar to the GPIS, the internal segment here, because both are tonically active, right? So the GPES is actively inhibiting, by releasing GABA, the subthalamic nucleus, okay? So it's preventing excitatory input from making its way to the subthalamic nucleus by inhibiting the cells here, okay? So this is inhibited tonically when the system is at rest, okay? When the system is activated, we have to imagine that both the substantia nigra pars compacta, right, which will release dopamine in an excitatory way, and the cortex, which will release glutamate in an excitatory manner, are both going to be triggered. When the pars compacta is stimulated, so maybe we'll draw in blue so we can follow what's going on. So when the pars compacta is stimulated, right, that excitatory output increases the inhibitory output of the medium spiny neurons, right? So this excitatory output increases this inhibitory output, okay? So now this tonic inhibition that was already present is disinhibited. So the brakes come off the subthalamic nuclei, right? And at the same time, cortical projections to the subthalamic nuclei release glutamate. So now the subthalamic nuclei is ready to release its neurotransmitter glutamate because it's active now, okay? And the result of its activity is to strengthen the GPIS. Yeah, strengthen it. That's kind of strange, right? So as the subthalamic nuclei becomes more active due to these cortical projections, it releases more glutamate, and the result of more glutamate release is a stimulation, greater stimulation of the globus pallidus internal segment. And we've seen what this does. This provides tonic inhibition to the VAVL of the thalamus, right? So we have an increase, I'm running out of space here, but we'll get the idea. Right, we have increased inhibition, right? So we're stimulating the internal segment to release more GABA and keep the brakes on the thalamus. This may seem unusual, right? These pathways seem to oppose each other, and they do, okay? But first, I just want to run through this one more time so you're understanding what's happening. When the circuit is stimulated, right, both the SNPC and the cortex are going to be active at the same time. We'll deal with what happens in the pars compacta first, right? It excites medium spiny neurons. Those neurons go on to release GABA. The GABA inhibits the external segment of the globus pallidus. Now this is important because the globus pallidus was already releasing GABA. So release of GABA, an inhibitory neurotransmitter, inhibits the GABA release of the external segment. This is disinhibition, right? If we have negative output and you add a negative, basically we now have a positive. We've disinhibited neurons of the subthalamic nuclei, okay? The second piece of this is that the cortex is releasing glutamate onto the subthalamic nuclei, right? So if we follow this pathway down, right, these projections into the subthalamic nuclei are excitatory. The subthalamic nuclei is then excited, and it releases glutamate onto the internal segment, right? And so this internal segment now produces even more inhibition, right? Typically, the globus pallidus internal segment produces tonic inhibition, and when we give it more stimulus, it produces even more inhibition via GABA, okay? So we'll come back next time, and we'll talk about how these pathways that look to unite can have opposing effects. And then we'll start to see where neurons in Parkinson's break down and what that does to motor output. Okay? So just to finish this pathway off, I know I'm a little over time, but I just want to make sure you understand, right? So inhibition of the VAVL complex of the thalamus, right? This inhibition inhibits motor output, right? Because the VAVL is inhibited, not much activity is going to flow toward the motor cortex. Okay, boy, that's plenty for one day.